Alright, hey guys, what's going on? So we're starting another playlist. I've wanted to start this for quite some time, and that's the main reason I've started this. This is going to be on artificial intelligence. Okay, so I've worked a chunk of my life on learning artificial intelligence, artificial neural networks, convolutional neural networks, and all the rest. And this is something that is going to hopefully accumulate into something that it could be like your go-to source on learning how to construct neural networks. My focus in this is going to be looking at how we can use these for big pharma, for drug discovery and so forth, but we'll touch on other examples as well. In the first few videos, we are going to be talking about just how neural networks work, the theory behind them, and then we'll get into the computer code and all of that. So without further ado, let's go ahead and get started. You can make sure to hit that like and subscribe button and become a member on this channel by just becoming a member. You can also support through Patreon as well. So with that being said, let's go ahead and get started. So right now we're really just gonna talk about the biology of neural networks. It's not really the biology. I've called it the biology of neural networks because we're gonna learn about neural networks. We're gonna learn about the nervous system and how the nervous system works. And so let's get started. So I decided I wanted to get this started because I worked on neural networks in the past, and I did a large part of my work in bioengineering working in neural networks. Mostly in MATLAB, but I'm sort of starting to get used to Python also. And part of the curriculum, again, was to learn the ins and outs of deep learning, and so learning the principles and the code and so forth. And so let's go ahead and get started. So broadly speaking, how does the brain work? Well, from a bird's eye view, the human brain takes in information from the outside world and produces behavioral or instinctual output, right? So we have 5 dimensions, roughly speaking, in our overall general lived experience. That is taste, hearing, sight, smell, and touch. Okay, all of these are going to be the information that all these senses send to the brain are going to be sent through nerve cells. And broadly speaking, we could say, well, we could put weights on all of these, right? So something could have a strong taste or a very weak taste. Something could be loud or not loud. Something could look a certain way. Something could have a very strong smell, for example. And all these weights are going to sort of tune how strong these input values are, okay? Then the brain is going to sum up all these weights, right? So this is the sum of all the weights. And X corresponds to the independent variable that's coming here. So this is X1, this is X2, X3, X4, X5, and so forth. Okay, it's going to put a function on that. This is our activation function, right? And then it's going to produce an output value. So what does all of this mean, right? So all objects in sort of in this very crude sense live in 5 dimensions, okay? Where each dimension is an independent variable. The brain places weights on each dimension according to the object and then acts accordingly. So this could be, again, some behavioral output. You see and smell a skunk, right? For example, and I just saw one a few days ago. And that gives you some sort of instinctual output, right? The instinctual output would be to run away from the smell, maybe. Or you see a bear, right? Instinctual behavioral output would be to run away from the bear. Or you see and you smell and you hear the baking of a pie or something like that. In that case, you're not running away from it. You're gravitated towards it, okay? And so you have this output. This output is your behavioral, your instinctual output. That's what the brain is telling you to do after it perceives the world around it, okay? Now, there's going to be a lot of things around you that it's going to siphon out as being sort of low on the hierarchy of important things, right? So I could have a cupcake in front of me. It could smell really good, and I could have my mic in front of me as well. I don't really care that much about the mic. I'm just focused on the cupcake. So the weights that are attributed to me perceiving the mic, for example, are very, very low, whereas the weights for the smell and the sight of the cupcake are going to be very high, okay? And so the input information coming from the environment enters something called the hidden layer. These can, roughly speaking, correspond to interneurons. Okay, now if you don't know what interneurons are, we're going to talk about them just a little bit. Again, this is not a neuroscience course. This is more of a course on artificial intelligence. But we're going to talk a little bit about interneurons, what those are, and so we can understand this from a biological perspective as well. So these nerve cells are going to send their information to the final nerve ending, which produces the output. Okay. Diagrammatically, this can be visualized in this way, right? So you have all your input layers. All your input layers are going to be fed into your hidden layers, right? So these are your interneurons. These are your afferent neurons, and then your efferent neurons are going to be the ones that produce the output. Okay. So, and in theory, all of these should be connected, but what actually is going on is that not all of them are connected. So let's take a look at what a more realistic one might look like, what a more realistic neural network might look like. So there are many different types of nerve cells in the brain. Roughly speaking, we can place them in 3 different categories. Afferent neurons, which are going to be these guys, interneurons, which are going to be these guys, and then the efferent neurons, which are going to be these guys. So the afferent neurons are going to be the things that take in the information. The interneurons, this is where the brain is, right? There's tons and tons, billions and billions and billions of interneurons in our brain. And those billions of interneurons have all these intricate connections, okay? And these intricate connections, well, they're connected to more interneurons, but then eventually some of these are going to be connected to efferent neurons, which send a signal back to whatever is producing the output, right? Okay. All right, so notice how not all the nodes are connected to each other, right? This node is only connected to 2 nodes, okay? And this is, in neurobiology, this phenomenon is akin to the Hebbian principle, or Hebb's rule, which states that neurons that fire together wire together. So weights are going to get stronger and stronger if there's more firing between them. That's really what this is sort of, this Hebbian rule alludes to, right? So this idea, I have this image here. You have this neuron, okay? This neuron, it could be an interneuron. So suppose it's firing more with this neuron, right? It's more in tandem with this neuron, less with this neuron. So what happens is that the connections between this neuron, they begin to dwindle away, become a little bit less important, okay, in this perceptual process. And so you have a stronger connection between, again, nerves that fire together and they wire together. So there's a stronger connection between nerves that fire more frequently together. This process strengthens connections between some neurons and weakens them in others, and is mediated by glutamate receptors at the synapse, right? So I studied glutamate receptors when I was doing my research in graduate school, and these glutamate receptors at the synapse, these things are located at the synapse, and they are responsible for recruiting more proteins to enhance the strength and the signal between neurons. Synapses act as high and low pass filters, or high pass and low pass filters, as they process electrical signals as they travel through the brain. So when we zoom into one of these synapses, so if I were to zoom in, say, right here, we actually, there's a whole new world jam-packed into these synapses. So just think about your brain for a second. Your brain is not a very big thing. It's encased in your skull. What is it, like 10 pounds or something? I don't know how much the brain weighs. But it's this thing. It has billions and billions and billions and billions of neurons, and these neurons are all connected to each other. Now the connections, they're not like in some sense, they actually are tethered to each other. Well, there's these proteins called connexins that will actually tether them. But there's this whole world where we have these the neurotransmitters being released into this thing called the synapse. Right, that's the thing we zoomed in on over here. These things are synapses. And the synapse, right, it's riddled with tons of stuff. We have sodium ions, we have neurotransmitter, we have other proteins in here, we have lipids, right? We have all these things, and how an electrical signal gets passed from one nerve to the other is we have these things here. Right? These things here are being spilled out from these vesicles in here, right? So these guys here, these are vesicles that come in from the axon. So these are vesicles that come in from the axon. They fuse to the membrane. They fuse to this membrane. I'll use my pointer instead. Via these proteins, these are proteins that actually grab onto the vesicles, and they fuse them with the membrane that they're attached to. And this fusion is going to cause all this stuff to spill out. Okay? And then we don't really need to know what the names of these proteins are. But we'll get into those later. Not in this playlist, obviously, because this is more of a biology topic than it is an artificial intelligence topic. We want to stick a little bit more with artificial intelligence, but I kind of want to give you people who are sort of more computationally computer inclined just a sense of what biology is like. And so these things are neurotransmitters. Once they get fused to the membrane, they spill out into here, and then when they spill into here, they're going to bind to receptors, right? So we have receptors here, we have receptors here, but when they bind to the receptors, we can see this one is bound to the receptor. It's going to allow for sodium ions to pass through. Right, so sodium ions are going to pass through. So this thing's going to open upon binding to the neurotransmitter, and then when it opens upon binding to the neurotransmitter, sodium ions are going to pass through, and that positive charge passing through the channel into the neuron, that generates an action potential. Okay, so it depolarizes the cell. So when sodium passes through, we're climbing up this peak here. Okay, and then other channels are going to open. Other channels down here, say, are going to open, and they're going to allow for positively charged ions, specifically calcium, to flow out. So flowing out, that's going to be here. So as we have sodium flow in, potassium flow out, and then we have this. We go way down here to these negative values, right? And then we get back to baseline, right? This is more of a neuroscience topic, so I won't cover this too much. We'll go over neuroscience in a separate playlist. But the idea here is that when we look at these neural networks, what we are neglecting, what we're going to neglect, is the intricate world between that's going on in these nodes. That intricate world, right? This is where neurons connect. Okay, and neurons connect at synapses. So these things are like synapses, if you will. And synapses, again, they are the things that act as things that will siphon out information. Okay. All right. At the synapse, so the synapse is as far down as we'll go in terms of in terms of going deeper into the details. Going deeper into the details is an enterprise for neuroscience, not this playlist. So we'll create a separate playlist on neuroscience. Artificial neural networks don't really have synapses like this. in artificial neural networks, the synapses are represented by each layer and the hidden layer. And as we've noted, there are places where these are places where filtering takes place. So we filter out information, what we think is relevant information, which eventually gets passed through another layer, and then to the output. And so there are many different types of filters, as we are going to discuss now, or not now, but in the next video. What I want to leave you with is that the purpose of this filter is to coarse grain information and to glean patterns from input data. Okay, when you look at a dog, for example, you are certain that there are certain attributes of the dog that your brain will pick up on, informing you that you're looking at a dog and not a cat. Okay, let's just look at this example up here. The challenge for machine learning and artificial intelligence is to be able to take in enough global properties of a dog to be able to accurately identify future dogs, but also accommodate for enough differences, enough differences so as not to make future errors. So let's go through an example. This is just a really crude example. But when you look at these images of paws, can you tell which are cats and which are dogs? This is relatively easy. This is a cat, and that's a cat. Everything else I put in here was dogs. But without me telling you this, could you have done that? Maybe, maybe not. Could a machine have done that? Well, this is what the enterprise of artificial intelligence is. What if I gave you the image of an ear to each of the corresponding images then? So this is, again, for the two cats. I couldn't isolate his ear, but this one definitely a dog. This maybe a cat, but I already told you it's a dog. But anyways, you might be able to now deduce which are dogs and which are cats. Okay, so this was relatively easy, but these are examples. But these are examples that we don't really typically work with, right? So my grammar here is a little bit off, but the point here is that we typically work with things that have a larger noise, right? Larger background. So the signal to noise ratio is it could be really bad. We don't want it to be really bad. Ideally, we don't want it to be as low as possible. But the data that we get is going to have some noise. Okay, but as long as we have some signal, as long as we can pick up patterns from the signal, then our artificial neural network should theoretically work. Okay, and also the data is also going to be more complicated. So instead of looking at paws, instead of looking at these very visual patterns, we're looking at paw and ear patterns. Well, in the context of drug discovery, we're going to be looking at cellular expression patterns, for example, from a microscope, from a confocal microscope, and that's a lot more complicated, a lot more data, right? Or clinical trials. The clinical trials might give us some data as well, right? So we can get an algorithm then. If we can get an algorithm to look for the right patterns and siphon out the right amount of noise, then we'll be able to predict what we're looking at. Okay, and this is the enterprise of artificial intelligence. Okay, so going forward, we are going to talk about the activation functions. We're going to talk about cost functions. We're going to be talking about loss functions, and then we're going to get into the code. Okay, because this in the context of when we're talking about artificial neural networks, what we want to do is fundamentally get to making some of these as well. So we're going to look at the code, but I want to get a lot of these other topics out of the way first, so we can focus more on the code later. Then we'll get into convolutional neural networks, and then we'll go from there. But with that being said, make sure to hit the like and subscribe button, and I'll see you guys in the next video. Bye.